So here's our Interdisciplinary Institute, BioFrontiers, in Boulder, Colorado, where we bring physicists, computer scientists, and engineers together with life scientists to engage in interdisciplinary work and teach our students in the same manner. What I'd like to talk about today is, well, look at this. Can anyone tell me how long the era of science lasted? She's asking the class. What is the answer? Until 2025, perhaps. So this tremendous movement towards anti-science, not just anti-vaccine, but anti-thoughtfulness, anti-facts, anti-data in decision-making, is something that I think we scientists have some responsibility for, and it's because we really haven't done a very good job explaining what we do to the general public, which makes it easier for them to then dismiss us. There are many reasons for this, but I think one of them is that jargon, the technical language that is so useful in the laboratory. I can walk into the lab and say, why don't you just CRISPR genome edit that allele into the endogenous locus in HEC293 cells, and the student knows immediately what I'm talking about. But that jargon is an impediment of us then reaching what we do to the general public. And so I have been going around the world talking about not just all of science, but the science that I work on, RNA research and medicine, to the general public. And I even published a book last year, which has now been translated into several other languages, to try to use no jargon to try to reach the general public. And I don't think it's had a huge impact, but I think we all have to do what we can in this regard. And my theme is that the 21st century is the age of RNA. DNA is still important. It's 20th century stuff. We understand it very well. It's a bit boring. All it does is store information. But the RNA revolution is still in its infancy, as you will see. So what makes conversations with the general public a little easier about this is that at least now most people have heard about mRNA. They know that when packaged into little grease balls, the mRNA that encodes the SARS-CoV-2 spike protein can be injected into your arm and allow your immune system to mount a response the next time it sees something that looks like that spike protein. So why was the mRNA vaccine not more celebrated than it was? Why was there some fear about it? And I think it was because many magazine articles, books, and social media talked about mRNA as a medicine, mRNA as a drug. And that makes people leery. It makes them uncomfortable. And instead, I think we should have said that mRNA is natural. It's in every cell in our body. It's in all the food we eat. It's essential for life. And we know how it works. It's not a new mysterious thing. We've known for 60 years that the mRNA is a copy of the information in the DNA, which then encodes the proteins that Francis Arnold has just talked to us about. So this is a wondrous technology, and I think that it's too early to tell how much of a paradigm shift in medicine is going to be caused by it, but I'm looking forward to the possibility of mRNA providing more effective flu vaccines, because you can go from knowing the strain of flu that's going to be present in the next flu epidemic to synthesizing the flu vaccine with mRNA very quickly, whereas with traditional vaccine methodologies, which involved hand injection of 1 million chicken eggs in the United States alone each year, it takes a long time to produce the vaccine. personalized cancer vaccines, because cancer cells produce abnormal proteins, and these can be used to train the immune system to be on the lookout for any cell that is producing such proteins. And these are in clinical trials now and are looking promising. And therapeutic mRNA to replace proteins that are mutated in genetic diseases is a much tougher problem, because we have to be able to make enough of the protein in the right tissue to be therapeutic. But it's exciting enough that scientists are working on it. Now, that alone, messenger RNA's wonders, might allow me to claim that the 21st century is the age of RNA. But that's not the whole story. There are RNA molecules that don't even care about the genetic code, that don't act as messages at all. They're called non-coding RNAs. And I'm going to very quickly just go through 3 examples of these. That RNA can be a biocatalyst. It can catalyze biological reactions. We call these ribozymes. RNA can bestow immortality, and RNA can control evolution. And this is the CRISPR genome evolution that we heard about earlier today. So how about the RNA being a catalyst? Well, when I first set up my laboratory in Colorado, I started working on what I affectionately call pond scum. But not just any old pond scum. This single-celled organism called Tetrahymena thermophila, because it had 10,000 copies of the same gene in every cell. And if you're a biochemist, you want to have lots of material to work on. And so most of you have how many copies of each gene in your cell? 2, right? 1 from mom and 1 from dad. So 10,000 is a big deal. And what we found out rather early on was that this gene was producing an RNA that needed to undergo RNA splicing, as Rich Roberts has told you about. But there didn't seem to be requirement for a protein enzyme in this reaction. We extracted the RNA very rigorously, and it still had this splicing activity. So although the textbooks would tell you that there absolutely had to be an enzyme, and we knew that all enzymes are proteins, yet there didn't seem to be any such material responsible for this reaction. So it took us a couple of years to pick the petals on the daisy and figure out whether there was a hidden protein or whether the RNA by itself was sufficient for catalysis. And the RNA was in fact biocatalytic, and this led to myself and Sidney Altman sharing the 1989 Nobel Prize in Chemistry, which I was a bit younger then than I am now. Okay. Secondly, RNA can bestow immortality. Wow, that's a huge thing. But it's only at the cellular level, not at the organismal level, okay? So don't get too excited. But this is the RNA-driven machine called telomerase. Now, the ends of chromosomes, the so-called telomeres, if there isn't some special mechanism to elongate those as the cells divide, when they get critically, they shrink with each cell division, and when they get critically short, cells stop dividing. And this is a good thing for most of the cells in our body, because you wouldn't want all of your cells to continue to divide after you're an adult. But it also drives cancer, because cancer cells have a way of reinventing the activation of this telomerase enzyme, and this allows them to be immortal. Now, how does this work? Well, as discovered by Liz Blackburn and Carol Greider, who got the Nobel Prize for this work, there is an RNA at the heart of this telomerase enzyme. It's shown here as the purple ribbon, and it serves as the template to direct the construction of the telomeric DNA. It adds 6 nucleotides, 6 informational units to the end of the chromosome, slips back and adds 6 more, etc. The protein that was responsible for this was found in my own laboratory. The protein that collaborated with the RNA This was discovered by a postdoctoral fellow, Joachim Lingner, studying, again, pond scum, but this time an organism called Euplotes aediculatus, which had an enormous number of chromosomes per cell, and therefore an enormous number of chromosome ends, about 100 million per cell. You have 23 times 2 ends per chromosome, about 100. This had 100 million. So this allowed us to purify the telomerase enzyme. And then because of the Human Genome Project, we were able to jump from this Euplotes organism to human, because everything is related through evolution. Now, the medical community got very interested in this because there was a mutation in the regulatory sequence of this telomerase reverse transcriptase gene that was responsible for reactivating telomerase in cancer, and it is the third most commonly mutated gene in all of cancer after KRAS and P53. Still don't have any therapeutics based on this basic science work, but people are still working on it, and it may come in the future. Now, finally, RNA can control evolution. This is sort of RNA's ultimate comeuppance against its parent, DNA. It can come back and change the code of the DNA, and this is the CRISPR gene editing revolution, which led to the Nobel Prize to Emmanuelle Charpentier and Jennifer Doudna in 2020. Now, it isn't quite as simple as the red scissors cleaving the DNA, but RNA is the key to its specificity. The RNA serves, shown here as the pink strand, serves as a guide, and only when the RNA matches the code along the DNA double helix does the CRISPR machinery lock down, and then the Cas9 protein, which comes along with the guide RNA, cleaves the DNA. And why is that interesting? Why is that useful, to cut DNA? It sounds bad, right? Well, it turns out that through the eons, all living systems have developed repair machinery that will patch up broken chromosomes, because broken chromosomes are a really bad thing, right? And so it stitches the DNA back together, and it's in that repair process that you can insert new sequences into the DNA and thereby, for example, repair genetic mutations which are deleterious. And this is already an outstanding research tool, but the medical potential comes not so much in deleting bad genes, but rather in correcting the wrong code that is present in faulty genes. And so these are three examples of how RNA, beyond being a message, can perform exciting and essential biological processes, medically useful processes, which allows me to claim that the 21st century is and will continue to be the age of RNA. And again, I think it's so important that we tell our stories in a simple way to the general public, and that's what I tried to do in my book that was published last year. Thank you very much.